Voy a hacer la presentación de Diego en español, si bien la charla va a ser en inglés, ¿vale? Bueno, es un placer tener a Diego Megias hoy aquí en el Instituto Blas Cabrera. Diego es actualmente, desde 2021, científico titular responsable de la recién creada plataforma de microscopía avanzada del Instituto de Salud Carlos III. Antes de ocupar esta posición, de 2009 a 2021, fue el jefe de la unidad de microscopía confocal del Centro Nacional de Investigaciones Oncológicas y previamente, desde el 2003, fue técnico titulado superior en dicha unidad y con anterioridad técnico responsable en el servicio de microscopía confocal de los servicios centrales de la Universidad Autónoma de Madrid, con lo cual, por lo que veis, tiene muchísima experiencia y cargos de responsabilidad. Diego Megias es doctor en biología y ha dedicado toda su vida profesional a la microscopía, acumulando más de 20 años de experiencia. Durante todos esos años ha completado su formación tanto en lo relativo a las tecnologías de adquisición de imagen como en su análisis posterior, mejorando el nivel de aplicaciones y tecnologías disponibles en la unidad y destacando tanto en el campo de la microscopía convencional como en sistemas automatizados de captura o high content screening. Su apoyo a los proyectos de investigación se ha reconocido en forma de coautorías en más de 100 artículos en revistas internacionales. En el periodo de los 2 últimos años, señalar la coautoría en artículos de revistas del máximo impacto como Nature, Cancer Cell, Nature Communications, Nature Cell Biology o Cell Reports, entre otras. Así que muchas gracias, Diego, por venir. Es un placer. Así que adelante, gracias.
Okay. First of all, I would like to thank you a lot for the invitation, for giving this talk. My intention is that I will try to give you a glance of things that are basically in the field of microscopy, latest advances that we are running. But if this is a kind of interactive talk, I would not mind about that. So if in case you have any doubt, any question, I mean, as soon as it's not stopping very much, because I know your time is precious. So, or after the talk, we can discuss as much as we want about anything I talk. So, as Javier says, I've been working in microscopy for more than 20 years, mostly in confocal microscopy. And yeah, I mean, as I said, let's see, just try to summarize a little bit what confocal microscopy is, and basically it's optical microscope, so we work with light. No? Okay. So the question is that confocal is based in fluorescence staining, so we need to label samples with fluorescence. So confocal is not possible if we don't have a fluorescence sample. And then we have much higher contrast and much more resolution than in conventional microscopy or bright field microscopy. So just remind you that we are trying to make things bigger in microscopy. So in any kind of optical device or optical microscope, we are trying to make things bigger. So this is what we do. We use lenses, we use different kind of lenses, complex lenses, that can increase as much as possible this resolution. So basically in optical microscopy, conventional optical microscopy, we are in a limit of resolution around 200 nanometers. So everything that is below these 200 nanometers is difficult to define and resolve. We will see in this talk that we can go beyond. But anyway, fluorescence is something natural. So it's something we use. This is this is oil. It can be bad olive oil, which gives you a kind of fluorescence in white, or nice olive oil, virgin extra, with this red fluorescence. So just to remind you that this is a kind of natural process, and we are just trying to use this natural process for our labeling strategies. So with fluorescence, we can have much higher contrast. We can have quantification, which is something that is not so easy if you work in bright field, like in hematocylin, eosin, or DAB stainings, and also allows multiple color imaging. This is pretty basics, but let's go beyond. In optical microscopy, we cannot distinguish the third dimension, so the volume. The volume is kind of blurry, focus or not focus image, but we don't know about volume. In confocal microscopy, what we add is a pinhole, kind of diaphragm, that is able to select the light that is coming plane to plane. So for every plane, we will receive a plane of light without disturbing on the sample, without making any kind of physical damage to the sample. Main application of this is to distinguish colocalization between these 2 or 3 or 4 markers we have in fluorescence. So for example, we can have green and red signal that overlaps, so it gives us yellow signal. Here there is full colocalization, so you can see here that both dots are in the same position, while in conventional microscopy, this would be exactly the same if we don't distinguish among these different sections. So this is one of the main purposes of confocal, to distinguish things that are really together in volume or not in the set plane. So these are the results that we can obtain with as optical sections from a mouse embryo. Here we have, as I said, 3 colors, blue, green, red. This is what we get, and this is what we can compute. So volume, 3-dimension reconstructions, in the way that we know exactly the structure, not only in X and Y plane, but also in the Z direction. Then we can distinguish for things that are in the same position in the Z axis. The question, this is a mouse embryo, by the way. This is us when we were really child, really small. Mouse, human is not really different. So in fact, here the red sphere around is what is going to develop the placenta, and then the green signal are going to be us, basically. In this case, mouse, but as I said, you can put together both and you don't distinguish. So this is a really big object for microscopy. Here we have one cell. So we can think twice that everything has 3 dimensions. So this is a cell in divisions. This is a metaphase. So we can clearly see all the chromosomes at the mid phase, and then microtubule spindle and centrosomes in white. So even for single cells, we have to take into account the Z slices, the Z dimension, okay, the 3rd dimension. Because things that looks quite close in 1 dimension or in XY, they can be separated in 3rd. Okay? Another thing we can do, as I said, we are working in microscopy, so we have fluorescence, so we can do multiple color imaging. So we can separate and we can combine, and then we can get, for example, DAPI signal, mostly is for nuclease. We can combine with different fluorescent markers, so we can add markers and markers. We can get every signal separately, which is something that, for example, in flow cytometry, they try to combine and compensate. We take pure signal without crosstalk. And by doing this, we can get, I would say, 5, 6 colors maximum. You can go up to 7 doing complex things. But to be honest, it's really challenging. So as soon as you try to put together as more than 5 or 6 colors, it begins, you will begins to have some kind of problems. For this, we are using time domain. Not just the spectrum, not just where is every fluorophore emitting, but also the time that takes the fluorophore to emit. Okay? So it's not if it's red and there are green, red, and blue. We can distinguish among 2 reds because one is slower emitting compared with another one which is faster, for example. So for this, you have to synchronize laser excitation with photon emission, okay? But then you can compute this time. And then you can distinguish, as I said, between a red signal that is emitting slower than another red signal that is emitting faster. Okay? Why I'm saying about this? Basically, it's because we can use this, for example, for whole scanning of a single, for a whole sample scanning. Then, something we can do. we can do 3 steps of acquisition in the acquisition. So we can do kind of super fast scan. Then this super fast scan can be done in the confocal. We can combine, as you can see, we can combine 3 lasers, 3 colors in the same capture, so we are much faster. And because we use the time domain, we can split those colors. If you don't do this, you will have a capture that takes around 60 hours. We are talking about 3 or 4 slides in the microscope in parallel, big pieces of tissue. So it's a long, long time of acquisition. If you apply this time domain, you can reduce this scanning time to 40 minutes. I'm talking about 2 days scanning, while in the other way, you can do it in 1 morning. Okay? So you can go very, very fast because you don't have to care about this crosstalk between colors. So you can combine these acquisitions in the same capture, splitting these colors by this time domain. So this is an example. So we are scanning tissue sections from lung. These are patients with different kind of treatment, immunotherapy, chemotherapies, and so on. So these are the low resolution scans. Then what we do is that we isolate here the regions that are more interesting for us. We acquire all those regions with higher quality, and the regions that are interesting here, for example, this kind of immune cores are acquired with a higher resolution. So we can go from macro to micro. So we can go from the whole slide acquisition, to the single cell resolution. I will stop just in case someone calls. Okay. So now you have one application. You can scan tissues with multiple colors, speeding up as much as possible this capture. Always you hear confocal is really slow, confocal is, you can push the system, and you can go to the boundaries of the system, okay? Nevertheless, here in this application I mentioned, we have 4 or 5 colors plus DAPI, so 5 colors plus DAPI, but the tissue complexity can be higher. So maybe we need more colors. Maybe we will need more colors. So for this, for cancer is the most obvious application, but immune system also is present in this kind of things. We have to try to find a way to stain more colors, to label more cell types. So what if we need more colors? As I said, it's basically possible to do it in one shot, 6, 7 colors. With the time domain, you can increase a lot the speed. It's okay, but if you need more colors, something you can do is serial staining acquisition. So the question, how it works? So basically, it's chemistry and microscopy. It's both things. So you have a kind of staining, then you take the whole image of your sample, then you remove the staining, and then you stain again. And then you acquire again, and then you destain, and then you stain. So you make this kind of cyclic staining acquisition process. I see kind of doubt about this. So the workflow is something like this. So you have your staining, your slice, then you do these 2 labels, for example, then you acquire, then you bleach, then you restain, then you acquire, then you bleach, and then you start back. So there are many approaches now. In our case, we are using an automatic stainer in the way that we can do this for many slides at the same time in parallel. And we are using conventional confocals or wide-field microscopes. Because in this case, you can have 2 or 3 colors. So you don't mind very much about crosstalk, because with conventional filters, you can remove those possible crosstalks. In case you need higher resolution, you can acquire those samples in the confocal as everything. So this is basically how the workflow works. So you have a kind of first acquisition just to detect autofluorescence. Then you can do the first cycle staining, second, and so on. You can do this antibody removal by different chemist protocols, chemical reagents, so heat. We are using this one, SDS and beta-mercaptoethanol. It smells really bad, by the way, but this is what happens with beta-mercapto. But it really bleaches or removes this antibody staining, these antibodies from the sample. You will say, okay, probably you're affecting the sample. Yeah, this is something we have to deal with. And then we have to look for the smooth point where we are labeling with certain efficiency, and we can remove without damaging too much the sample. Just a kind of example of something we detect when we did this kind of assay. So this is first round of acquisition. So this is one labeling, first round. Then we remove this label, and then we label with red. And then we remove this, and then we label with green. And then we remove this, and we label again with green. You probably notice these scratches, okay? Or things like this hole here. So this is the damage that comes if you do serial labeling with this kind of methods. There are much softer methods. There are commercial systems available for doing this, which removes most of these kind of artifacts. But it also reduces the flexibility, and they are really expensive. So we are trying to do this at the Spanish way of doing things. So yeah, so basically we are doing this, and it's working pretty fine, to be honest. So except if Murphy's law appears, in most of the cases you will see that you have a lot of regions to see the overlap between among all these colors without these scratches or without this damage. Sometimes it will happen that this damage appears exactly in the region of interest you want, but this is something we have to suffer. So here you can see that after several rounds, we still are able to retrieve the signal from the first round. So this chemical erase of antibodies is not damaging so much the antigenicity of the tissues, okay? You can see clearly here, this is CD68, then we bleach, or we remove this CD68, then we add SOX2, then we remove, then we add CD206, just to see if we can see green signal in the CD206. And we didn't saw any signal, because this is kind of nuclear, this is kind of one cell type, and it's completely different cell type. Then we remove this, and we acquire again SOX2, and we compare the regions, or the signal, among second round of staining for SOX2 and fourth round of staining for SOX2. And we again apply CD68, and we recover basically the signal. And here you can see the overlap between round 2 of labeling for SOX2 and round 4 of labeling for SOX2. So you can clearly see a part of kind of different, and this is because the tissue is not exactly the same. You can clearly see that the nucleus that were positive in green are still positive in red. So we retain the epitopes from those labels. So these are things you have to do to set up your multiplex assay or your spatial biology assay, and then you can begin to run this through different markers through different rounds. Okay. With this, we are now in 16 colors plus DAPI, so we are talking about 21 colors. You have one blue, 3 visible, then we remove another blue, 3 visible. We use the blue signal to overlap the different rounds of staining. Okay? And obviously, as I said, you can say, okay, yeah, you have some kind of damage. Yeah, okay, again, we cross the fingers and we hope this damage doesn't appear in the region of interest. We have a whole brain to look for those interesting cells. In this case, it's also because the tumor is here located. So this gives a kind of different morphology to the tissue and different resistance to the chemical agents. But it's working pretty fine. So we are working this now with this basically as a routine. So you can clearly see round 1, 3 markers, round 2, 2 markers, round 3, 2 markers, round 4, 2 markers. And it's exactly the same region. So in case you have samples from patients, in case these samples are precious and you cannot do serial sections as if you're crazy, or in case you want to clearly see if one cell is related with another cell as a single cell level, this could be a kind of alternative, right? So yeah, I'm not going to stop very much on this. You can read about this. It's something that's already published, and there is a kind of lot of papers and journals showing about this. And remember, you will need always to combine this with single cell analysis. So image analysis is also really important, not just labeling, not just acquisition, but image analysis. As you can imagine, you can get the single cell data, and in most of the cases you have to go to machine learning or artificial intelligence algorithms to group, cluster, because as soon as you go beyond 2 or 3 colors, humans, or at least I'm a little bit lost. It's really difficult to make, okay, negative for this, but positive for this, for negative, but you know what I mean? So it's kind of complex. And then, depending how deep is the organ, depending how deep is the organ, you will need to open certain parts of this organ, or you can go to skin, which is much easier, or also you can do endomicroscopy. So this is basically the setup we had at the Seminario at that time. So we have a kind of stereo microscopes, anesthesia, injections, and so on, and all this amount of things for putting a kind of poor mouse in the microscope. So, but basically you can see it's again Spanish approach. So with magnets or rubber bands and kind of electricity devices like this. But basically we were able to keep the mouse still alive and with a kind of compatibility with microscopes. So this is a kind of psoriasis event in skin, and hopefully this runs. Yeah, this is lung. In fact, the movement, this movement is not because the blood, it's because of the air pressure going in and out from the lung. And you can clearly see a green signal from GFP and blood vessels. Okay. Let me tell you, if you, I mean, you don't notice about how fast things happens into a body until you see it. I mean, it's super fast. You don't imagine, because you are used to work. I was used to work with cells in culture, tissues, and so on. In the body, everything goes super fast. So we are injecting in this assay, we were injecting tumoral cells at the eye, and then we detect those cells at the lung. It took less than 1 minute to arrive those tumoral cells to the lung and engraft into the lung. Less than 1 minute. I mean, you have the time to click, click, and click. So is this fast? So you have to adjust the speed in the acquisition, so you can see again blood into a tissue. So you can see here, if you don't adjust, you don't get, you don't get single cell detail. But if you go as fast as possible, you can clearly see single erythrocytes flowing through the blood vessels. The main problem we have in intravital microscopy is the scattering and the absorption of the light. So this is what makes us impossible to go deeper into the tissues. Probably you hear or you saw recent paper in Science about making transparent human or mouse skin. This is going to be a clear revolution. As soon as you can make transparent the skin of an animal, a living animal, and go inside, at least for science, I'm not sure about commercial applications, but at least for science, for sure, it's going to be a kind of revolution. Because in most of the cases, the problem you have is that you lose this focus point because of the scattering or the absorption. So it's quite difficult to get signal from deeper layers on the tissue. Super difficult. You need super high efficiency, and then you get kind of blurry or no image. So this is why those chemical reagents for clearing tissues appear. The idea is, okay, if I have a problem because of the scattering and I have a problem because of the absorption, let's remove this from the tissue. In this case, and this is the difference with the Science paper that appears, is in fixed samples, so it's not in living animals. Okay? If your model, your animal model, is transparent, like zebrafish or some kind of microorganisms or organoids till a certain point, you can work without clearing anything, and you can work in vivo samples. But as soon as they get color, it's much more difficult to get. But you can clearly see how you begin with a brain, a mouse brain, and then you reach kind of certain point where the sample is completely transparent. So you pass in confocal from a depth of 60, 80 microns, depending on the complexity of the tissue and the density, you can go to 1, 2 millimeters. So in most of the cases, you lose the focal distance, the working distance of the objective, and you still can get image. So you can clearly go deeper into the tissues. Obviously, this is a kind of long process, so you have to remove and wait for days and make the labeling and so on, but it's possible, and it's giving super good results. You have, just in case you're interested, just take a review or something, because you have a lot of chemical approaches for doing this. There are some of them are compatible with fluorescent proteins, some of them are not. You have to choose, okay? And depending on the organ also, you will have some kind of variations. But basically, it's removal of lipids. This is what you are going to do in most of the cases. And this is basically what the people for the skin transparency are doing. So you can see a kind of skin piece, a piece of skin here and here, 2 wells. This is in our lab, and then what happened before and after clearing. Here you can see that you don't see any fat, and you just see the hair of the tissue, and the whole tissue is completely transparent. Obviously, this is something you have to prove. You can compare here 2 kind of clearing methods. This is a control sample. This is UDisco. This is cubic. Cubic is compatible with fluorescent proteins. UDisco, not so much. But you can clearly see here, here there are cells. Here we have the same layer of cells. Here we have much less cells, by the way. But then we can pass through these cells. We go to the muscle, and we reach the blood vessels behind, which is something we don't see in the control. Okay? So in the control, we just finish, before the muscle, and this is my fault. And while in the other ones, you can go beyond this muscle and reach those blood vessels. So you go much deeper into the tissue. Applications, we run this in skin, as I mentioned before. We run this in lung. You can see this is, in this case, is immunostaining. So you can see clearly alveoli, you can see cells, positive cells in green, positive cells in white, sorry, and some autofluorescence. So just imagine you have a really rare population of cells into a tissue. And now you go with conventional approach, which is section and section and section and labeling and section and labeling. For a 3 millimeters tissue organ, you have 20 micron section, 10 micron section. It's super difficult to find these rare events. While in the other case, you have the full lung. And you scan the full lung. So the level of sensitivity, the level of resolution, the structure, where are those cells, is still preserved in these clearing methods. This is kind of lymph node. And yeah, this is the comparison of this lymph node without clearing and with clearing. So you can clearly see that you go deeper. It's not only for mammals. We do this in flies, for example, for other animal models. And this is head that we, fly head that we put in the social networks. And you can clearly see that you go deeper into the fly head. Briefly, just to mention a little bit about samples throughput, we are clearly biased. I mean, I've been discussing with people about microscopy, no, but this is subjective. You have really few number of cells. How you can be sure that your data is right? Okay, the microscopy is not like this anymore. You don't have to spend hours in front of the microscope. Obviously, sometimes you have to, but in most of the cases, automation is taking the place. So you can clearly see that Ramon y Cajal here is just sit down here and do something with a microscope. In fact, he doesn't have a sample here. So this is the typical thing that happens for us. But basically, the idea is that every single microscope, as soon as it's automated in focus, obviously wavelengths, and stage, you can run multi-well plates, you can run serial or mosaics of tissues, you can do a lot of things. Now this is automation, so you need to have a kind of clear workflow. So it's basically biology is behind, or materials, depending what you want to do. And you have to follow this kind of pipeline to get kind of robust workflow for automation. You are not going to see every single image you acquire. So you have to trust in the data. Okay? So this means that you have to be really strict in the procedures you follow. There are some, I'm not going to stop very much on this because of time, but kind of super nice example I always showing is about cell cycle. We can use DAPI signal, which is an intercalated reagent in 80 pairs in DNA. And here, if you ever work with flow cytometry, you probably notice this kind of two peaks. This was obtained with a microscope, and this was obtained with a flow cytometer. So we can get exactly the same parameters. So 2N, 4N mitosis, and we go back. Nice thing about microscopy, automated microscopy, we can run this for 96 well plates. And we can get this in less than 1 hour. And we can clearly see if the two peaks are affected, like in this case, or have a kind of wild type behavior. This is another example for foci nucleoli. Nucleoli in this case, this is a kind of nucleolar disruption assay, or for example, for mitochondrial membrane potential. So here you can acquire and quantify mitochondrial potential. You can see translocation of different fluorescent proteins here in this case, for example, to ensure that the nuclear transportation elements are working. or pathways are working, or if they are excited or not. Obviously, if you don't have a kind of microscope, you cannot run these assays. Because for a flow cytometer, this is the same green than this. So you need to know if it's in the nucleus or if it's in the cytoplasm. So with this kind of automated acquisitions, you can run clearly proliferation assays. It's super easy, mitotic index, cell cycle, and many, many other parameters. I'm just trying to give you ideas and so on. The only thing is about automation is try to keep this as much simple as possible, because if you have certain variability in one label, then you have to add, if you add a second label, then you have another variability source, then you have a third label, you have another variability source. So in most of the cases, I would recommend to make a kind of super simple assay to scan as much samples as possible and then get a kind of second round of selected cells or selected samples where you will run a kind of second labeling, much more complex with higher resolution and so on. Okay? Anyway, if you are interested about this, you have my email or whatever, we can discuss. Not very much about cell dynamics. We move from morphology, microscopy, conventional microscopy is morphology, expression localization. We can observe dynamics. We can see cell dynamics. So you can use this kind of syringe pumps to make a kind of flow when you interact with the cells in the way that you other chemist or any kind of induction or kind of treatment, you want to see what happened with the cells. And you will see online what happened in the cells, because you are here adding any kind of treatment and you're watching at the same time the treatment is added, what happened with the cells. So you can add this for, in our case, we are using 6 channels. So you can have this kind of syringe pumps, the 6 channels, and you can here, for example, see how long the glucose stimulus is appearing in the cells. And then you can compare silencing, treated, wild type, whatever. Okay. This is for natural killers. So these are tumoral cells, and then we add those natural killers, and you can clearly see that we can track those natural killers, and we can clearly see how they bite and how they surround tumoral cells until they kill it. So you can also distinguish about the efficiency of those immune cells and see this and watch these super killers, these supernatural killers that appears in the population. In this case, it's about platelet addition, so you can clearly see tumoral cells and to detect this platelet addition to those tumoral cells. So this is something you can see online, and this is something you can do to compare all those different samples. Last but not least, about the latest advances in resolution. Remember, resolution is not how small is the object I can see. This is sensitivity. Is if I'm able to solve or divide 2 objects that are really close, or if I am able to get the right size for the right object, for this object. So it's not that you don't see virus, it's that you don't know how many viruses you see. Okay. So resolution, the main problem in resolution is kind of diffraction of the light. So as soon as you pass through a tiny hole, or you are trying to observe a tiny object or super tiny object, then you have this kind of diffraction. And this diffraction is exactly the same that happens in the sea. So as soon as we have a wave and we try to pass that wave through a hole, then we are creating a second wave. And this second wave is what limits the optical resolution. So with conventional optical microscopy, you can reach, as I said, 200 nanometers resolution. So for cell biology, it's super nice. But if you want to go beyond and you want to go a little bit to higher resolution, then you have problems. They were so sure about this that they write the formula which relates wavelength and resolution in stone. And if you write something in stone, it's because you are really, really sure. This is not a paper, okay? The question is, it was a super resolution diffraction barrier. And it was relating wavelength and resolution. So if you use visible light, you have a kind of super narrow window of wavelengths, so resolution is limited. And this was really true until certain years ago, I would say 15 or something, or maybe 20. I mean, first we're in the labs, now we're present in commercial systems. In our case, there are several approaches for super resolution. In our case, we are following the STED, which is stimulated emission depletion microscopy. And then what you are able to do with this kind of systems is that you switch off the fluorescence temporarily in the way that you can block the fluorescent emission around the center of the focus. Okay? What does this mean is that if you have a kind of excitation spot which is limited by diffraction, this is still valid. The stone is perfectly there. Nothing happens with the stone. The stone is still present. The only thing is that as soon as you put a donut around of a second laser which is able to block the fluorescent emission, the emission of the fluorescence, then you can get only the fluorescence from the center of this excitation spot. So therefore, you are increasing the resolution of the system, because you don't get the fluorescence from the full spot. You are getting the fluorescence only from the center of the spot. This depletion laser is coupled with excitation laser in the way that as you are using a confocal system, so this is kind of easier system for putting into a confocal. So you can combine those two lasers, and then you are getting only the center of the signal from this, and you scan the whole sample. So this is a kind of video explaining this, just to let you understand this. So here you have focus limited by diffraction, so you have these two spots, and then when they pass through the focal point, if you don't have enough resolution, what you have is a kind of one single spot. And this is the problem. So again, if this is a virus, if these are two viral particles, I will see them, but I don't know if there's 1 or 2 or 1,000. While if you put the STED system, you are blocking the signal until those dots appears into the center of the point, on the focal point. And then you're splitting these two dots. Okay? So this is something we did in the lab. So this is nuclear pore protein. And then you can see the difference between confocal and STED image. So for example, here you can see this spot is coming from 3 spots or 2 spots or whatever. Okay? So you can clearly go beyond the resolution limit of a conventional microscope. And this was not so easy 20 years ago. It's still not easy, I would say, but at least it's something you can run. As I said, there are different ways for doing this. You can do STED, you can do STORM, SIM, structural illumination microscopy is another alternative. Depending on the sample and the system you use, you will have some limits, but you have many approaches for this. And also, this super resolution can be achieved also in the set, not only in XY, but also in Z, because this donut, this depletion donut, can be formed in XY, but also in the Z direction. So then you are improving the resolution not only in XY, but also in Z. What does this mean? This means that if we have an image in confocal, we have an image in STED, and we can clearly see that we are able to distinguish much easily the source of this fluorescent emission. And then you can count, you can see morphology, you can do things that moves microscopy closer to electron microscopy. Okay? I'm not saying it's the same. Okay? Electron microscopy is beyond. But we can fill the gap a little bit better between optical microscopy and electron microscopy with all these possible applications for living cells, multicolor, deep imaging, and so on. Just to remind you, for example, about viruses and so on. So with the STED system, you can, commercially available systems are around 30, 40 nanometers, I would say. If you are lucky and your sample allows, you can go beyond, but most of the cases you will go for, as I say, 30, 40 nanometers of resolution. And this begins to allow us to see single viruses. Okay. Again, this is super resolution microscopy. I just want to mention a little bit about expansion microscopy, which is basically a kind of super different approach. Physics, microscopy, put lasers and lasers, expensive systems and so on. Then the biologist appears and they say, okay, if something is really tiny and then we don't have resolution, what happens if we make the object bigger? If the mouse is as a cat, it's easily, you can observe much easier. And then it's in most of the, this kind of protocols appears not so long ago, I would say 10 years. And this is exactly what they do. They take cells and then inflate the cells. Yeah. Super nice papers. The what? So you are saying that you can make one cell looks like 2, 3, 4 times, 10 times bigger? I was really skeptic about this, but then we tried. And if you follow the recipe, if you follow the workflow, you can make a cell looks 10 times bigger. And if you do this, and you can see that this is also possible not only in XY, but also in Z, this is a kind of nucleus before and after expansion, you can clearly see how the structure is preserved, because this kind of expansion is isometric, so it works in XY but also in Z. So you are basically separating the objects, and as soon as you separate the objects, you can solve them. You can see if there is 1, 2, or 3, or whatever. Again, probably you don't trust me. This is science, so this is 20 times objective, zoom factor 2. This is what we saw. This is what we saw after expansion. In this case are centromere signal and heterochromatin signal in red, centromeres in green. So okay, my doubts about this, okay, I see a bigger cell, but maybe the proteins doesn't work like that, Yeah, I mean, colocalization is not changing very much when we compare this with this. Obviously, you lose a lot of cells, so you have a lot of problems with the workflow and so on. It's difficult to do. But basically, you can count more single centromeres in the expanded cell compared with the non-expanded one, which actually means that we increase the resolution. I'm not saying this is better than the other thing. Probably this is much more complex in terms of protocol and so on. But just to let you know that there is people expanding cells and then doing super resolution. So the expansion of the expansion. So these things are done now. And this is final image of this. So I hope I convince you that we have more penetrance, more resolution. We can do automated acquisitions. We can see dynamic cell dynamics. We can couple this with microfluidics, interaction with the samples, and so on. So come to the dark side. We have cookies. Thank you.
Thank you, Diego, for this interesting talk. Now we have some time for questions from the audience.
Okay, so thank you for this super interesting talk. I mean, I've learned a lot. Also, my brain like kind of crashed with some of the things, but anyway. So I'm very interested in how do you image scattered tissues without optical clearing. Okay, we're very interested in ocular tissues, particularly sclera, which is super scattered tissue. So we have been doing multifocal microscopy on clear tissues and things like that. Adapted optics and that kind of approaches doesn't work because scattering cannot be corrected. So I wonder if you know another approach up to trying to image biological scattered tissues without chemically modifying it? Or
There is people, I mean, there are two possible answers for this. I would say if you have scattering tissue and you cannot, let's say, modify this scattering, problems are there. I mean, the scattering is there, you cannot block the scattering. It's also true that you can, there is another kind of microscopies where they are trying to measure the time. The idea of this is that as soon as you get the time, I mean, if you have a lot of scattering, okay, it means that the photon is delayed by the scattering to reach the other side. So then, by using this kind of time domain, you can get the time that the photon takes to arrive to the detector as another parameter. So it's kind of complex thing, okay? A part of this, without going in this, because probably this means a kind of different system and a kind of different approach, here it's like organoids or all these kind of things. So the idea of this is that, okay, I have an organoid and I don't want to touch the organoid and I want to go into the center of the organoid. You have a lot of scattering because all the different layers, the organoid is, for example, if it's a brain organoid, you have dead cells, then you have multiple layers. Here, it's a matter of compatibility with the system. I would say, first, you can go to red colors as soon as possible. Are you trying to look for label cells, or you just see the kind of structure?
we want to see like collagen fibers and maybe fibroblasts.
Okay, for collagen fibers, you have this kind of second harmonic generation, third harmonic generation. You can try to do this with multiphoton. Yeah, this is with time, yeah.
Oh, sorry. Yeah, thank you.
No, I mean, I'm not sure about which is the right answer for this. I think I would say, if you were my user, I would say, okay, it's super important to go to this level of sclera, or you can go shorter times, get smaller ones, and then try to guess. I mean, what I mean is that, okay, let's imagine this happens in 1 month or 2 months. I don't know about your sample, okay?
Yeah.
Let's imagine 2 months. Okay, and then in 2 months you have this 150 microns limit, and then you can go beyond, and you want to, okay, then you can get this 150, and then try to correlate with what happens earlier in time, where you have much smaller object or problem. And then as soon as you can establish this correlation, and then you will see, okay, this kind of, I hope it's mouse or humans?
We have
both?
rabbit, pigs, and humans.
Okay.
Together at the same time. But
Depending on the animal or the animal model you're using. I mean, the question is that probably, I'm just guessing, you can tell me whatever. The question is, if you wait 2 months to see what happens in pig or in mouse or something, or in the rabbit, then probably you can see, for example, okay, I have a super big problem, in this, which 2 weeks ago, when I had much better optical compatibility, I had this kind of a structure. And then try to establish kind of model about what happened at earlier times compared with later times. You know what I mean? In the way that then, the question of this is that, let's see about this kind of skin treatment and so on, probably it will expand a lot the kind of applications, because in fact what they do is that they match the refractive index of the skin. Yeah. And probably is what what will happen, I mean, as soon as you can put it in the eye, which is something. But I think it will go in this direction, and this is why I think this kind of approaches will be super important. But so far I would say that if the microscope doesn't reach signal beyond these 150 microns, even considering that you are using a multiphoton system, this means that the light doesn't go there and goes excite and goes back. Obviously, in these kind of things, it's again about labeling. If you put a lot of signal, probably you can get some signal back. And yeah, try to make it as much compatible as possible. I'm sorry very much for not giving, okay, you can use this thing and that's all. That's all. No, it's kind of super complex
to me. If you would have given like a very, that's very easy. That would be killing for me.
Yeah, I don't think so. I mean, we are talking about optics, and this is kind of limits.
Okay, thank you so much.
No, you're welcome.
That's another question.
Okay, thank you. Just a stupid question. You have
No.
shown a lot of setups. I was wondering how many confocal microscopes do you have? Do you share confocals between different setups, or each setup has a confocal?
We have a facility right now. Those images came from the Seminario. Nowadays, we have 2 confocals, 1 and 2 wide field systems, and 1 for holographic imaging. And we bought the HCS system for this year, so probably this will be the platform, the full platform. Main problem if you get a lot of confocals is that you have to keep running those confocals. So as soon as the time passes, it's super expensive. So yeah, I think it's depending on the number of users and working, the time that the people needs to approach one of these systems, then you have to. But as I said, if you begin to automate acquisitions, and I don't know if you are doing this means that the system is running overnight, is running without people. So you can get a lot of samples without so many hours. So you can make a system works 24 hours, 7 days. So you can reduce a lot the queue on users list. But yeah, right now what we have is a kind of a Stellaris to Thunder SP5 DMY 4000 and Nanolith for holographic system.
But it is easy to adapt. Because you have some like microfluidics, you have some like in vivo imaging of mouse.
Everything is coupling this into the systems we have. So they are not dedicated systems. So as soon as someone comes with microfluidics, then we put the syringe pump, then we put all the tubes and capillars and so on. Then this means that we have to book the system 1 hour in advance before the assay, and then we take this time for preparing the system. For intravital, we dedicate the full day. Okay, so then when we block the system, we block it for the full day.
Okay, perfect, thank you.
Because you don't want to disturb with the animal now, we have a delay and now.
Okay, thank you.
Thank you, Diego. It's been really nice, and we'll get to see you tomorrow once there. I was just wondering, in the multiplex staining and so on, one of the major problems has been always the autofluorescence that is removed mostly with Sudan black. And then when you were doing some staining, you were using an anti-fade. But if you do that, it's kind of incompatible with doing several cycles.
Yeah, I really hate Sudan black, I'm sorry. So there is another kind of, for lipofucsin and autofluorescence in brain tissues is super.
Yeah.
So if you have one of these tissues is when I would recommend to use confocal, because it's not that you block the fluorescence, the autofluorescence, it's that you can dedicate a channel to see if it's autofluorescence or not. And you can modulate the detection bandwidth in order that you have a channel that says, okay, everything that appears here is autofluorescent. And then this allows the image analysis and removal of these autofluorescent artifacts. The question is that, for example, when we try Sudan black and we tried sometimes, there was a kind of super nice idea, which was to put a sample into a transilluminator for DNA and put it in UV for whatever, but at the end of the day, the autofluorescence was still there. So I think that this much better because of the Sudan black treatment probably, at least in our hands, the antigenicity of the tissue is reduced, the signal that you get is much lower than comparing with another one without any kind of treatment, depending the tissue nature and so on. But I think it's much better to use these autofluorescent channels for autofluorescence in the way that they distinguish if the signal you have is autofluorescence or not. And we are working like this for many years, and it's working pretty fine.
Okay, and another thing, in the several cycles, you make use of the antibody removal, because you use trees, either you go to pH, acidic pH, or but you always use SDS. SDS is clearing the tissue.
Yeah, in fact, it's a kind of reagent that is used for tissue removal.
You strip it, but you remove. You make it clear.
Yeah. in most of the cases, this means that you are removing, I would say, more lipids than proteins, which actually is not. And all these things are trying to keep the structure. So there are different approaches. We are following this kind of clearing methods. There are other approaches for cyclic staining, where they simply use conjugated primaries, and then with a chemical reagent, simply they uncouple the fluorophore in the primary antibody. So it's much softer for the tissue. So they put a kind of residue in the antibody, between the antibody and the fluorophore, and then they do a chemical treatment and they remove. This is kind of commercial thing, and the problem of this is that you are forced to use primary conjugated antibodies, so you cannot use primary and secondary. We are following this other approach. First, because there is something that you have to, I didn't mention, but you have to keep in mind, is the multiplex assays means 16 antibodies, or 20 antibodies, or crazy people is doing 50. Every antibody is around 1,000 euros, multiplied by 16. I mean, this is the main limitation in multiplex technology, I would say, is the price of the reagents. If you go for conjugated antibodies, probably you need more primary because you are not amplifying the signal. So it's a kind of expensive technology, I would say. Not for the technology, because it's a microscope and autostainer, but because of the antibodies you want to. So it's a project that you have to run if you need it, not just for fun. But apart of this, I can tell you that now we are running 2 projects, one in mouse and one in human, and they are working pretty fine. So it's super important to define which are the primaries that are you going to use, to check those primaries are not affected by SDS treatment or by this clearing that you are making into the tissues. But as I said in expansion microscopy, everything can be wrong, but it's working. So as soon as you have controls, and then you check the same, for example, SOX2 that we made in round 1 and compare with SOX2 that we made in round 8, and we can clearly overlap the same cells, this means that the antibody, at least the antigenicity or the epitopes are still present. So depending again on the treatment and so on, you have to double check in your tissue sample, but for sure it's possible, and there is many people doing this. I can tell you that if you couple this with tissue microarrays, for example, when you have a slide with 70 samples, 80 samples, and there is people doing this, you have 80 samples, 20 colors, then you need computing and so on, because it's quite complex to use.
Thank you very much.
Yeah, so thanks for the talk. I was just curious, because there's probably people in the audience here that it's more, would like have more interest into applying confocal microscopy into the analysis of materials.
Mm-hmm.
So could you comment a little bit on how some of the principles that you explain apply?
It basically, in this case, I would clearly recommend to explore also reflection, light reflection. You can get confocal images from light reflection. This is something, for example, in case you use nanoparticles, iron nanoparticles, magnetic nanoparticles, you will have some kind of reflection from light. Also, if you want to see smoothness in a surface or something like that, you can use reflection, because the light will change reflection as soon as it passes through different points. And then you can get the microfractures and so on with a microscope, optical microscope resolution. So yeah, I mean, everything related with nanoparticles. You can use a fluorescent signal for labeling these nanoparticles. You can use reflection, as I said. And you can, for example, something we do is that we inject those nanoparticles with microfluidics, and then we clearly can define the time that takes the nanoparticle to enter into the cell, for example. Or if the efficiency of entrance, if it's 100% of the cells with the nanoparticles inside or not. I'm not sure if when you mention materials, you are talking about nanoparticles or surfaces or...
Yeah, actually I was thinking more on surfaces, but
I mean, we did kind of, for microfractures, I worked a long time ago with this kind of dental implants and they check if the cells are adhering to these dental implants and the surface, the smoothness in the surface. Also, if some kind of surfaces have, as I said, microfractures. And also, for example, there are kind of chips that later are going to be used for cells and then can measure, for example, the pressure of the cell, how much pressure. I mean, it was super curious. It was a kind of chip that changed in wavelength emission, yeah, when depending on the pressure the cells was doing onto the chip. And then you can guess this kind of things. NSET, which is kind of FRET, but because nanoparticles is also possible. And yeah, I would say as soon as it's fluorescent or is you can get reflection, and in most of the surfaces you have reflection. I would say even in soil, you have reflection. It's perfectly possible. I mean, you just need to add a thin layer of things of the material and then check surface, clustering, aggregation, or I mean, diffusion. As soon as you put it, yeah, all these kind of dynamic assays are possible. If it's fluorescent, you can use confocal. If it's not fluorescent, you can use reflection. And let me tell you that, for example, in case of reflection, you have different reflection depending on the nanoparticle for different wavelengths. So you can get one kind of particle reflecting in 555 laser line, and another, and in the same sample, you will have another kind of particles or surfaces or aggregates which are reflecting in a different wavelength. So reflection is not as fluorescence, but you can find different reflection from different samples in a surface. So I would sure recommend to try this reflection with different laser lines. In this way, you just put the detector not in the fluorescence, but behind the laser line, and then you can get those different reflection and compare. And probably you will see kind of different behavior in surfaces, for sure for structure, but also for composition. And you can get the autofluorescence from these nanoparticles or surfaces, which actually can be also different.
All right, thank you. Any more questions? All right, so let's thank Diego for his talk and discussion, and also we would like to give you a little gift from the Institute as appreciation for your visit. Thank you.
Thanks a lot. I will open now. Uy. Estaría guay. Ah, mira, ni tan mal. Así me acuerdo.